Well, welcome everyone. Welcome to all our attendees. I'm very happy you could join us for this post-COVID-19 rebooting with research panel discussion on viral detection and mitigation. This rebooting with research series looks more closely at cutting edge University of Waterloo based research that's making a difference and leading to the establishment of companies. Before proceeding further, the University of Waterloo would like to acknowledge that we live and work on the traditional territory of the neutral Anishinaabe and Haudenosaunee peoples. The University of Waterloo is situated on the Haldimand Tract, the land promised to the Six Nations, that includes 6 miles on each side of the Grand River. So hi, my name is Bridget Maloney. I am the Managing Director of Jedi at the University of Waterloo. Jedi is the gateway for enterprises to discover innovation at Waterloo. The university is proud to host this webinar series. This is the 5th today in a 6-part panel series. COVID-19, as we all know and have experienced, has shaken our world, challenging societies and altering life as we know it. But from this crisis, opportunities are emerging, calling us to action as we prepare to reboot from this COVID-19 lockdown. How we work and behave, how society and the economy will function, and how we will innovate and educate will all change. So plotting the future for Canada, your enterprise, and yourself is complicated and challenging. So we're very happy today and through this series to have our researchers and university partners sharing their research and their perspectives. So just a couple of quick housekeeping items. If you have a question for our panel, please send it via the WebEx Q&A by clicking Q&A in the bottom right corner of your window and selecting all panelists before sending. This event is being recorded and will be uploaded to the official UWaterloo YouTube channel. So now I'd like to introduce our moderator and panel, and I will just show a quick slide so that you can all see who are joining us today. So I would like to introduce our moderator, Professor Sushanta Mitra. Sushanta is the Executive Director of the Waterloo Institute for Nanotechnology, or short form WIN, and also a professor in the Department of Mechanical and Mechatronics Engineering. So Sushanta, over to you.
Thank you, Bridget. Good afternoon, everyone. We have an exciting panel here with industry as well as top-notch researchers from Waterloo. And one other aspect of all these researchers are they are not only doing excellent research, but also able to translate that research into value-added products and services. So this is really an exciting afternoon. We are looking forward to it, listening to their perspective on this COVID-19. So I will start requesting maybe a very short, brief introduction of each panelist. So I'll start with Ryan. So Ryan, please.
Sure. Thanks, Sushanta. So hi, everyone. My name is Ryan Denemy. I'm one of the founders and the CEO of Nicoya. So I'm a Waterloo alumni. I was actually the first class of nanotechnology engineers to graduate from UW. So many of the other panelists were actually my professors back in undergrad. So it's great seeing them all again. After finishing my undergrad, I did a master's also at UW in Patricia Nieva's lab, and I worked on developing low-cost biosensors for point-of-care diagnostics. So I spun that research out into my own company called Nicoya right after graduating. That was in 2012, and I've been working on that ever since. So what we do at Nicoya is we make scientific instruments that use a biosensor technology called surface plasmon resonance, or SPR. And this is used by scientists to better understand diseases and develop drugs to treat these diseases. So essentially, it measures how well a drug binds to its potential target, and then it can be used to screen hundreds to thousands of different drugs or vaccine combinations to find the best one out there. And so what we've done is use nanotechnology to greatly reduce the cost and complexity of the instrument so that every single lab in the world can have access to this powerful technology. So we have customers all over the world at top academic institutions, government labs, pharma companies, biotech companies. We've had quite a bit of success the last couple of years. We've raised tens of millions of dollars from venture capitalists and the government. We've grown to over 60 employees that are usually situated at our office downtown Kitchener. And we've got lots of exciting stuff going on, so I'm really happy to be here today.
Excellent, Ryan. So now may I request Kareem, please.
Perfect. Thank you, Sushanta. So my name is Kareem. I'm a professor in engineering and also the executive director of the Center for Bioengineering and Biotechnology. I also happen to be a co-founder and CTO of KA Imaging. It's a company that I founded along with Amal Karnik, who's the CEO, and one of my former graduate students, Sina Ganbarzadeh. This was probably about 6 years back. And the company commercializes the work that I was doing in the lab, which was related to X-ray imaging. So one of the most interesting things that KA Imaging has done today is it's created the world's first portable dual energy X-ray camera. And why that's useful is because these portable devices are really good at visualizing pulmonary disease. So if I'm looking, for example, the progression of COVID infection, which turns into a pneumonia, then this type of device is very useful. And because it's portable, it becomes even more useful because now you can clean it. You can take it to the patient. You can take it into a long-term care facility. You could take it to an indigenous facility if you needed to, in low-income and middle-income countries. And even in regular hospitals, you could take it into the ICU. You could take it into emergency rooms, and that's somewhere where this type of diagnostic has not been seen in the past. And so this device has just recently been submitted to the FDA for a 510(k) approval. So we hope maybe by closer to the end of this year, we have the approvals and we're able to, or we have the clearance rather, and we're able to start getting it into the clinical sites where it can actually be used.
Thank you, Kareem. So may I now request Rod to introduce yourself. Rod.
Hi, thanks very much, Sushanta, and thank you to Jedi for the invitation. My name is Roderick Slavchev. I'm an associate professor from the School of Pharmacy. I also have two companies that are spin-offs out of the UW and out of the research that comes out of my lab, the first being Metaphage Bioceuticals, located in Toronto, and the second is Theraphage, which is actually taking part of Velocity right here on campus. A lot of the work that we work on is the generation of new therapeutic platforms, all with some spin or basis on the genetics of bacteriophage, which are bacterial viruses. So in one platform, we actually are very heavily working within the gene therapy field and trying to redefine how this works, moving this more towards a redosable and titratable system with new vectors that we like to call genetic medicine, and looking at genes as actual medicines that could be used for personalized medicine. That's Metaphage Bioceuticals, and more recently Theraphage, which was launched just 8 months ago, is specialized in the use of bacteriophages themselves as new types of immuno-oncology agents or as cancer vaccines. So based on this research from both of these, the genetic medicine as well as use of bacteriophages, I'm working currently with Dr. Marco Coyne and with Dr. Emmanuel Ho, and we are trying to address the COVID-19 crisis with a new vaccine and trying to make use of both of these techniques as well as therapeutic platforms of their own. And that's something that's well underway. We are one of the 102 contenders right now currently working toward a global vaccine candidate, and things are currently, it's currently going well, so we hope that that continues into the near future. Thanks very much, and again, pleasure for being here.
Thanks, Rod. Really appreciate it. May I now request Shirley to introduce?
Thank you, Sushanta, and thanks to Jedi for the invitation to join the panel. My name is Shirley Tan. I'm a professor of chemistry and also associate dean of science for research. Prior to joining Waterloo, I actually had 3 years of industrial experience in addition to academic experience. My research focused on developing intelligent materials and devices to meet critical medical needs. And in particular, my lab developed wearable and portable devices for distributed healthcare needs. Now, in 2016, a company was spun off from to commercialize the technology from my lab, and it's called the Nano Diagnostics. It's positioned as an in vitro diagnostic technology innovator, and the main platform is a portable point of care testing, essentially for immunoassay. And we're hoping now to leverage this system for COVID-19 testing. Thank you.
Thank you, Shirley. And now, Alex. Alex, please.
Hi, everybody. I just want to make sure. Can you hear me clearly? Okay, perfect. So I'm Alex Wong. I'm an associate professor in the Department of Systems Design Engineering at Waterloo. I'm also the Canada Research Chair in the area of artificial intelligence and medical imaging. And my main focus has been on the operationalization of scalable and transparent artificial intelligence, especially for machine learning and deep learning. And a number of different startups have spun up from our research lab, including Darwin AI, which is a local Waterloo company focused on accelerating deep learning development. And some of the projects that I work on have been largely related to operational medical imaging analysis and diagnosis, with one that's more topical related to building deep neural networks for the detection of COVID-19, as well as risk stratification for COVID-19.
Thank you so much, everyone. So to kickstart the panel discussion, maybe I'll first talk about this recent announcement where Nicoya is one of the 4 Canadian companies to receive a significant federal funding to develop portable COVID-19 diagnostics tests. So Ryan, could you please tell us how you see Nicoya's role in fighting COVID-19?
Yeah, absolutely. That's a great question. I think fundamentally, our mission at Nicoya is to improve human life by helping scientists succeed. So since COVID started, we've been playing a pretty active role through a number of different mechanisms at Nicoya. So, our first priority when this really started was making sure that we could still support our customers. We have a number of customers that are in virology and doing research on vaccines, specifically aimed at COVID-19. So we wanted to make sure that we could still support them and help them get their research projects underway. So, we launched a number of initiatives, including free support, a really detailed educational webinar series put on by our customer success scientists. We put out a number of application notes that are showing how we can use SPR in the fight against COVID. And we've also partnered on a number of NSERC Alliance grants and helping a variety of different projects move forward in that sense. So, those are some of the things we've been doing already, but, while that was happening, we were also keeping a close eye on how the landscape was developing with respect to diagnostics for COVID-19. And it quickly became clear that having a really large capacity for testing was going to be critical to being able to contain the virus and ultimately, ease a lot of the social distancing measures in place in a way that would be safe and effective. So, we've been kind of looking at it and trying to understand, how Nicoya can leverage its technology and expertise in order to help solve some of the issues around testing and diagnostics, because we do have a team and a number of platform technologies that are very applicable to the diagnostic space. So one of the big issues that we saw when the epidemics are getting worse and worse is that the turnaround time for testing was a major problem. So right now, if you're feeling like you might have symptoms of COVID, you go and get a test and it's going to take 2 to 5 days to get that result. So, in between that time, if you think you might have COVID, you've got to quarantine yourself, which means, you can't go to work. It's going to affect your family life, your employees, your employer. It's a challenging situation to be in. And you might not have COVID, but you have to assume you do. And if you don't assume you do, then you might actually have it and you could go out and be infecting a lot of other people unknowingly. So one of the things that would really help contain the spread of COVID is if we had a really rapid test, that could be done at home, self-administered, a simple saliva-based test that could give you results, in 20, 30 minutes. And that's what we've really been focusing our efforts on, and happen to also line up really well with the challenge that the government had put out. They were looking for tests that were similar in nature to that, to be able to move testing into more of these decentralized point of care settings. So that's when we proposed and have been developing our technology called Atlas. So it's basically a low-cost saliva test you can perform at home, connects to your smartphone. Within 20 minutes, you can get the results on whether or not you may have the COVID-19 virus. And it can obviously easily push these results out to the apps like were announced today by the government for better tracking and tracing. So we're really happy that we've been able to get some support to work on this. Obviously, lots of work still to do, but we're to get it out to the market as fast as we can.
Thanks, Ryan. Just to follow up on this, since you are talking about this, maybe if you can provide a little bit more background and or maybe throw more lights into how you see this rapid diagnostic platforms are evolving for the fight for COVID-19, Shirley.
Well, thank you for the question. I think you already had a key word in your question, which is rapid, and every minute counts. And if you look at the testing evolving from January to now, I would say a couple of things. One, the turnaround time has been shorter, both through technology improvement and also by simply building up the capacity in the infrastructure. And another thing is we're looking at the more diverse targets. It used to be only RNA-based tests available, and still in Canada, I think, or Health Canada approved tests are RNA-based, but we're looking at more and more research and commercial efforts to push for antibody testing. And another aspect is, like Ryan mentioned, is the different samples that can be used for testing to provide more convenient test protocols and better user compliance. So saliva is a new arrival, and researchers start to understand where viruses can be found, where antibodies can be found, and their timelines, and how they can predict for the kinetics of the infection, and etc. Now, if I looking from this point forward, I think for research, we're looking at more and more demand for multiplex testing for one, improved accuracy, which is what we suffered actually, still suffers from the existing test. And the second thing is more information about history, prediction, and probably assist in treatment. Another thing is more connected testing, smartphone paired with on-site testing. So it will allow more, they call it geospatial mapping. So this is really helpful for policy setting purposes to look at outcomes of certain strategies in place, and also enable very fast response in terms of isolation.
Thanks, Shirley. So another aspect of the diagnostic is looking at imaging, and what Karim mentioned is about looking at soft tissue images and so forth. So Karim, from K imaging point of view, how you see K imaging is able to respond quickly to the needs of COVID-19 in terms of treatment and also diagnosis of the disease itself. So Karim, please.
What we have noticed in our conversations with clinicians is that the accuracy question for PCR keeps coming up over and over. The existing tests today, in the worst case, may be only 30% accurate, and in the best case, maybe 80 to 90%. But there's a big range in the middle. So what we find, a lot of the protocols that are being put into place in emergency rooms, in hospitals and departments, is when a patient comes in and they're suspected of COVID, they will do a PCR test. Sometimes they will do two to try and improve the accuracy, and they will pair that up with a chest X-ray. And the idea then is that the chest X-ray, because the disease starts off in the lower parts of the lungs, every so often the chest X-ray can pick up the disease before it actually shows up on the PCR. So in fact, there was some research that was published early in March in Europe, where they actually did a small study, and what they found is that in about 10% of the cases in that study, the chest X-ray caught an abnormality in the X-ray earlier than the PCR actually showed up as a positive. And then 2 days later, the PCR actually showed up positive, and then we knew it was COVID. So what KE imaging has done now is we've basically improved upon regular chest X-ray. So what KE imaging, like the company has got, is we've done something known as dual energy X-ray that allows us to separate the soft tissue and the bone image in addition to the regular X-ray. So we can see the soft tissue where the COVID-related disease, the pneumonia-related disease, is more visible, and the idea is that in places where you're doing PCR tests, the X-ray is used as a complementary method to catch any of the cases that PCR doesn't. The other interesting piece here is that while PCR is quite a valid solution in high-income, wealthy economies, it is actually not extremely feasible in low-income or middle-income countries, or places where you have really, really high population densities, but you may not have the income to support it. So for example, the WHO recently is now is recommending the use of chest X-ray in the management and the even in the screening of COVID-related disease. In fact, with a number of our conversations with the radiologists out of Tanzania, they indicated to us that PCR is essentially not accessible, or they just don't have the capacity to be able to work with it. And so they've been reliant exclusively on chest X-rays. And of course, they're not as accurate. We know that because the chest X-ray cannot diagnose COVID. It can only screen for COVID because it gives you an idea of a lung abnormality. But even still, it has that ability to predict the abnormality, which is being used right now in low and middle income countries to actually catch suspected COVID patients.
Thank you, Karim. So COVID-19 also taught us a very good way is seeing that the medical innovation in Canada is happening in a very rapid space. And when we think about medical innovation, the first thing that comes in our mind from a Canadian perspective, insulin and banting. So maybe our next banting is sitting with one of us here as a panelist in terms of maybe Rod, your discovery in terms of vaccine and how you see it as TheraPage and working towards the fight of COVID-19. Could you elaborate and tell us what are the innovations you are looking forward and where this is going forward in terms of vaccine and therapeutics?
Sure, I'd be happy to. That's a tall order to live up to, but yeah, definitely one of interest. I'll have to say that never have I ever seen so much research done in the vaccine space in such a short time. This is truly an exciting time, and not to take away from the grimness of COVID-19, but what is actually happening here on the scientific front is rather revolutionary. Currently in Canada, there is a lot going on, as there is, of course, globally. In the World Health Organization, there are 102 vaccines currently that are listed within this landscape. We are one of those. And this is a combination of some of your more regular and conventional approaches to vaccines, as well as some of your much more regular and what might be or might show to be particularly relevant to the SARS-CoV-2 virus, but also may be particularly pertinent and perhaps more therapeutic in terms of what their potential is within this space. That being said, there are some bottlenecks on the regulatory issue. Now, it's worth saying that the typical vaccine takes probably around 20 years to get to market. And in some cases, for instance, for HIV, it's been 35 and we're still not there. So there are complications and it is virus dependent. SARS-CoV-2, as is SARS-CoV, is actually a very tricky virus in that space where you might see some of your less regular types of vaccines that are actually making the biggest difference, although they may take longer to get to market. Currently within Canada, I'd say that we're pretty well positioned. There are a handful of players at present within the universities as well as some of the private sector. Most of your major universities do have a contender for vaccine going forward. And we even have one in phase 1 from Halifax and Dalhousie at present, which you may have heard of, which is one of interest and is a partnership with a Chinese company. So there's definitely a lot in the works. As I did mention, there is a little bit of trickiness to this virus, and I think you'll see some of your non-conventional approaches that really start to make a difference. In our case, we tried to do as much in-depth research as possible to try to account for some of these differences and try to attest. And this actually will relate to some of the diagnostic testing. And I had a few comments listening to some of the excellent types of research going forward that I think will play into even the diagnostic application of this. Vaccine-wise, what we're working on is something that we are actually combining a few different types of efforts. The first being something that's called a DNA vaccine, which means that we try to deliver some of the viral components or the antigens to the cells of the lower respiratory tract, which is the primary site of infection of SARS-CoV-2, and to within these cells actually generate something that is a viral shell, something that is completely harmless, has no genetic material within itself, but kind of synthetically or simulates a type of infection within the actual site of infection. And there's a lot of new data to suggest that trying to mimic a route of infection as closely as possible is perhaps the best way to generate the most pertinent type of an immune response. And this is both your humoral, in terms of the antibodies that are presented, as well as the type of cell-mediated response that's generated. And further to that, because it's normally inhaled as an infective route, we are also using an approach that is an inhaled route or nasal route, which has a couple of advantages in that it tries to stimulate type of immune cells within the nasal cavity that's rich within that type of an antiviral response that we're looking for, as well as getting this synthetic infection, as we call it, or the simulated infection to the actual lower respiratory tract and route. Now, one of the biggest problems that I see within Canada at present and that we're encountering at present, and this is in teaming up with Theraphage and Metaphage as spin-offs as well to try to expedite this process, is taking 20 years of vaccine design and moving it down into something as short as 12 to 18 months. In Canada, we have two particular points that are lacking, and I think that you'll probably find this across all the contenders that are trying to get to clinic. One is manufacturing. Most of these facilities we will look elsewhere, usually to the States, to try to provide the scale that's necessary, particularly for a global level vaccine. And the second is on the clinical side, because we need, particularly for phase 3, we need exposure. And this usually requires frontline workers to be able to provide that exposure and that safety element and efficacy element versus a placebo. So those are the two areas that I see particularly as a bottleneck. And although this is a phenomenal global union that's occurring and collaboration, the problem is that your final vaccines or your final formulations that occur are usually a combination of a variety of different countries, which, although great from a collaborative perspective, puts a few bottlenecks into the actual processing for one country to be able to supply what's necessary to their own people in a very quick and expedited fashion.
Well, we definitely need the vaccine yesterday, but I think we have to wait maybe a few more months. Who knows? Maybe from your innovation, this will accelerate very fast. So till the vaccine is available, we are our economy is opening, and therefore social distancing, contact tracing, and so forth is becoming more and more critical than ever. And the data is so much central to our decision-making process. So this brings to Alex. So Alex, how you see AI, machine learning playing a critical role in terms of early warning, as well as how, as a nation, we prepare ourselves into the second phase of this pandemic? Alex, please.
Yes, so that's a very good question. So I could see the role of AI, machine learning, deep learning in a wide variety of different ways to help not only with the management at the hospital level, but also at the city level. So just coming off what Kareem was talking about before, in terms of the importance of chest x-rays as part of the clinical workflow for COVID-19, one thing I want to add to that is, in addition for detection purposes or screening purposes, one one thing that it offers that PCR as well as antibody tests don't offer is risk stratification, which is the understanding of the severity levels as well as risk levels of a particular patient. And that's something that is critical. And so on that front, myself and my colleagues, as well as my collaborators at Darwin AI, we've been working on AI models for taking, as Kareem mentioned, chest x-rays that are actually quite difficult to interpret for radiologists, especially early on, and especially for frontline workers who might not have the same level of expertise, and leveraging AI to give them additional insights of likelihood of COVID infection. Again, as Kareem mentioned, it's not diagnostic, but it's there for the initial screening, but also give them a better hint as to the level of severity, because that's also very hard to judge from a human level. So this is also very useful information for controlling, for not only personalized care for individual patients, but also for managing supply chains, hospital resource management, and so on and so forth. So there's a lot of different ramifications with having AI in the loop in the clinical sense. But as you mentioned, what about for, I guess, COVID management in general? One thing that people have started to look into is, can we leverage it for social distancing assessments, as well as for understanding the critical factors when there might be issues. So one particular area that I'm particularly interested in and also have expertise on is more on computer vision, AI, video analytics. And so there's been studies, as well as myself, where we started exploring, can we leverage computer vision to understand the locations as well as the density of individuals, and then being able to see whether people are actually keeping social distances, and when they don't, whether actions to be taken or whether a particular venue should be reorganized to enable better social distancing. So those are some of the other facts. And last but not least, there's been a number of different studies out there right now that is trying to leverage AI and the wealth of information that's being collected from the different sources, be it from clinical sources, be it from social media sources, so on and so forth, and using AI to parse through this large amount of data and get a better understanding into where are some of the key hotspots for COVID, as well as predicting whether or not there might be a increase or a decrease in cases moving forward. And so these kind of predictive analytics can really give, I guess, our policymakers as well as our healthcare workers early warning signs, as well as ways to figure out how to actually control spread, as well as how to mitigate the situation.
Well, thanks, Alex. We can clearly see, Darwin AI and K imaging can work together in this kind of excellent efforts and really fighting COVID-19 for our country as well as globally. So Kareem, maybe you have some further inputs to this effort that we just talked about in terms of partnering Darwin AI with K imaging and other aspects of machine learning with imaging.
I was just going to add, Sushanta, that Alex raised a really good point with regards to X-ray not only being used for early stage as a complement to PCR, but also in the monitoring of COVID infection. And one of the places where we see this a lot is patients where we intubate different set of, and many of the time it's unfortunate, but those patients who go on the ventilator, many of them don't have the ventilator. That can actually longitudinally measure the progress of disease. We could determine optimal times when the ventilator should be used. The other place where this type of technology could be also very, very useful is in our and so these patients are highly vulnerable, and there's a challenge moving them from the hospital and then bringing them back. That we have been. One of these X-ray machines enabled with AI in a long-term care facility, in every long-term care facility, so that the patients can be evaluated and assessed in situ at the bedside, really at a point of care, if you imagine. And you can see that this would require policy changes, but it could also lead to some very nice outcomes in the future, where we could avoid a lot of the situations that we have right now.
Thanks, Karim. And I think as we are seeing overall, be it imaging, diagnostics, and other efforts, the government is definitely putting a lot of funding to it, to fundamental research and also even early stage translation. But often, even today, there's an article in Global Mail which suggests that where as a country we are sometimes lacking is time in kind of translating that quickly into public sectors in terms of taking it in a much more scale up way. And here we have Nicoya. So Ryan, who is able to translate some of the university driven research and taking it up in a scale up way, along with other panelists here as well. So Ryan, how you see that university industry, particularly in this case, Waterloo, Nicoya relationship building up overall, and also in particular in the context of fight for COVID-19?
Yeah, I mean, like, as a company that, founded by, Waterloo alumni, we went also into the Velocity program, early on. And, those types of partnerships, I think during the really early stage of a company's existence are just so critical. And I think the Velocity program and a number of the other programs that are located in the region here have really helped to make it easier for companies to start and to get through the first couple years, which are typically some of the most challenging for a business. But we also got our very first funding through a FedDev UW grant. So we definitely wouldn't be here without that funding. So all these programs have an impact and make a difference for individuals and companies trying to translate research out of universities. It's certainly not an easy thing to do, and it's as much infrastructure and support as can be put into that. I think we'll continue to see great companies forming out of University of Waterloo and other institutions. For us, we've had a pretty long history with hiring Waterloo co-op students, which is very helpful in the early days of a company, because you can do this with fairly low risk. The students can be there for 4 or 8 months. Exactly how much it's going to cost the business, and you can get quite a few government grants to reduce the cost even further to the business. And there are some really smart, talented students that you can get to help move particularly technical projects along. And on top of that, we've gone back and hired multiple co-op students full time once they've graduated. And that also is a huge benefit to the business, because it's a lot less expensive to go through a recruitment process when you've already worked with somebody for 4 or 8 months and you already know they're a great candidate. So that's I think a testament to the program, as well as the quality of students that are coming out of some of the programs at UW. We have a ton of engineering and science students from UW that we've hired full time. We've also had many collaborative projects with research labs at UW over the last 8 years, and ongoing now as well, to help support the development of our Atlas diagnostic product. So being able to leverage specific expertise and skills, and making that happen really easily, I think, is important. You don't want to have a lot of barriers in the way. When you have companies and professors trying to work together, you don't really want there to be a lot of bureaucracy and red tape getting in the way, because both groups just want to move forward and get moving, especially in times like these, where we need solutions rapidly. We can't be stuck in negotiating contracts for 6 months. It's just it's not practical. We're not other schools, other companies, other countries will quickly move a lot faster than we will if we get caught up in those types of things. I think the other thing too is just looking forward, there's a lot of opportunity for the tech companies like Nicoya, University of Waterloo, and the other post-secondary institutions, and the hospitals, the doctors, the clinicians in this region to really like form some stronger partnerships and work more closely together. I think this pandemic has really showed us that we have a ton of talented people here in the region, and if we can just get those connections working together a little bit faster and a little bit sooner, we'll have a better avenue for piloting health tech related innovations, getting feedback from clinicians, getting access to patient samples for testing. I think there's a ton of opportunity there, and that's definitely I think would be a great area of focus for us and the other stakeholders going forward.
Excellent, Ryan. Happy to hear that Waterloo is working with Nicoya and other industries to really lead our effort in terms of fighting COVID-19. Internally within the university, also research institute like the Waterloo Institute for Nanotechnology, where we have members like Karim, Rod, and Shirley, who are working within the institute as well to make this happen. So maybe Karim, since you are involved in WIN, being a thematic lead of therapeutics and theranostics, and now leading another institute, which is the CBB. So how you see that what Ryan just mentioned about the energy and the effort that are happening between external engagement, how you see that can also be leveraged in terms of internal collaboration, partnership across institute, for example, between WIN and CBB and things like that. Karim.
Within the institution and beyond, so that we can help translate all these new powerful technologies as quickly as we can. And the institutes should really work together. I think that's a great opportunity for being able to make sure that we have some solid very quickly. And of course, making sure that we have a place where the clinical innovations can land. Linkages and partnerships that we need to put into place so that we're kind of ready to capitalize on all the great work our researchers are doing.
So I think definitely speed is critical in terms of bringing these efforts together. So for speed, we talked about it earlier as well. Rapid diagnostic is critical, one aspect of it. I know, Shirley, your lab as well as your startup is working on some of those rapid diagnostic platform as well. So if you have something new to share with the community, that will be exciting to hear as well.
That's a good question. I would like to give a little bit of history and what my lab and my company has been doing over the past 5 years or so. So we're trying to develop nano biosensors and trying to answer a couple of unmet needs in the medical diagnostic field. One thing is, can we manage to do testing outside of the hospitals, outside of the labs, and really provide good quality testing data? That means we have to reduce costs, we have to reduce the amount of reagents needed, and that means we have to remove the need for trained professionals. And also another thing is, can we have detection technology that actually sensitive and accurate enough that it provides the same quality of testing data that you would have in a lab testing? And another thing is, once it's outpatient, and can this testing be adaptive? For example, for COVID testing, we know that the viruses mutate very fast, and whatever we are working on right now is most likely will be applied in future, different virus strain or changed. Can the platform be more adaptive? So those are the key questions we are asking ourselves and saying, can we do this? Can we provide detection technology actually address those unanswered questions? Now, the platform we come up with is essentially a handheld system, very similar to the one that's for glucose testing. That's about the only, I would say, chemical marker that's constantly be used by patients or users. And we're saying, like, can we create an equivalently convenient, easy to use platform, but really target the biomarkers maybe 1,000 or 1,000,000 times lower in concentration that we cannot detect them in the past? And can we really have also multiplexed testing to have antibody and the virus testing at the same time? And all those things are hopeful. I can't say it's ready now, but this is the direction we're heading to. And one thing that we've addressed so far is with the biosensors made of nanomaterials, the very, very miniature. It's really easy to create an array of the sensors to read the idea for multiplexed testing. They have high sensitivity, so really idea for pushing the detection limit. And another thing is we've come up with a platform essentially miniaturized whatever in the lab, like the electrochemical station, to a handheld system, essentially equivalent to the size of smartphone. So it is a system solution that we're trying to tackle from the instrument side, from the material and the sensor side. We're also tackling from the analytical chemistry side. And more recently, we're also involved machine learning, trying to improve our power in terms of more accurate analytical prediction, and also improve our capability to be hooked online.
Well, thanks, Shirley. As you have pointed, I think one of the challenge we are seeing for COVID-19 is also there is the variance of this. The structure can change, it can evolve over the different population, different demography, and so forth. So that brings the question about the vaccine as well. So, Rod, how you see as a global community we should able to tackle this evolving challenge with its structural change and mutation, possibly, of this COVID-19?
Sure, absolutely. Actually, if you don't mind, I have a couple of things that I just wanted to say regarding a couple of the former comments from Kareem, Shirley, and Ryan. One with respect to the bureaucracy. I think this is a really rich time in that it's one of the times only that I'll remember where you see industry and academia really aligned with the same goal. And I think that we have a lot to learn here, and hopefully we can bring back rather than, being deluged with rules. Also to that, I mean, having a much more efficient process that's goal-oriented, but also the student part. One thing that we're doing at Theraphage is to run students through this industrial cycle. And very often, especially in basic science, students do not have the opportunity to see the end result of what they're doing because they're so far up the supply chain or up the pipeline of of the value chain of what's occurring. I think it's a really rich experience for them to really see how what they're doing can make such a huge difference. To what Shirley said on the multiplex testing, and something I wanted to get to is that SARS-CoV-2 is actually a very strange virus, and there are a few others like it, like Zika and dengue and respiratory syncytial virus, whereby the first infection can actually prime individuals for subsequent infections, and the second time round can actually exacerbate disease or facilitate infection by your own immune response. This is due to non-neutralizing antibodies that can be formed, or even cell-mediated response in that respect. And I think from a multiplex testing, if we could also not just look at the diagnosis and being able to test individuals that have COVID-19, but also the predisposition to some of these other antibodies that may increase their severity for the next serotype that comes along. That's, I think, a really, really important point. And also something that then, as I move this into vaccines, is very relevant to vaccines. It's not just about durability. Durability here can actually also be the problem. Because if we design vaccines that specify an immune response that generate these non-neutralizing types of antibodies, as well as this potentially immune-enhancing type of an immune response, then we are setting individuals up for potential downstream health issues and exacerbation of disease rather than its prevention. And that is a really important point to take into account with this virus, and particularly in the vaccine design, in that a lot of your more conventional approaches may not be the ones that work here. The second part to vaccines as well as to SARS-CoV-2 is the concept of lung inflammation. A lot of the vaccines that have been tested for SARS, again, the most conventional approaches to this have generated lung inflammation and eosinophil accumulation, which is not a good thing and definitely can actually cause more problems in severity rather than a solution and potential protection for individuals. Now, if you look at the actual sequence of SARS-CoV-2, there are a lot of sites of conservation. It remains to be seen whether or not these are primarily immunodominant over time, but these conserved sequences you can expect to be recurring over and over again. And believe it or not, SARS-CoV-2 does not mutate that rapidly, especially compared to something, say, like a retrovirus, or even to influenza. It might be somewhat similar in that respect. It's more where it sort of mutates. If we have a natural selective pressure toward these non-neutralizing type of antibodies that can exacerbate disease, that is a powerful selective pressure that we need to be able to screen for, and definitely has to be taken into account, probably ahead of anything, for the safety of the types of vaccines that are going forward. So again, when I said this at the beginning, I think that this safety component is something that may be a little bit unique to this situation, but something that has to be taken with importance, or with great importance. And I think even from the diagnostic perspective, a lot can be done to try to screen for these.
So in terms of therapeutics versus vaccine, who is winning this war currently, or they are equal footing now?
Well, it's hard to say. I mean, to be honest, SARS-CoV-2, we don't really, all in all, know that much about it. And we don't have a lot to go on, really, historically, from it either. But from the studies that have been done, even on SARS-CoV-2, there's a suggestion that both are obviously important. One can provide potential solutions immediately, albeit one of the biggest issues is the lung inflammation. So even some of the sort of inhaled types of approaches to try to treat may not be so effective because of mucus accumulation that may limit the ability to target the sites that are the sort of basis of these approaches. Vaccines, there are, I think, a lot of potential that come from some of your more novel approaches. Some of these, for instance, mRNA approaches to the receptor binding sites, and these, I think, are very unique and have great potential to move forward. In the end, it's hard to say. I mean, really, we have to watch and we have to learn. But one of the greatest issues that I see right now is that we have a balancing act between safety and efficacy. And obviously, we're trying to push the efficacy envelope as much as we can. And it's really important that we pay proper attention to the safety components as we do so, to make sure that we are staying on top of where the potential issues can be.
Well, definitely a complex problem, and that will keep all of us busy, definitely. Indeed. For the next few months. So as seems to be, this vaccine development is quite complex. It's predicting these kind of signatures are also very challenging. So I'm wondering, Alex, is there a way machine learning or AI can play some role in terms of providing some sort of a pathway towards maybe a targeted vaccine development and so forth? So Alex, if any thought on that process?
Sure. Just a couple of thoughts on that. So what I found, at least with a lot of recent, I guess, advances, is there's now been an increased focus on leveraging machine learning, particularly deep learning for pharmaceuticals, as well as for vaccine development. So there's been individuals look into how AI can be applied to identify particular molecular markers, targets, and so on and so forth, and seeing and simulating whether or not they would actually have a strong effect or not. And so that has actually been very interesting. And there's also been some recent work by other colleagues where they've been looking into building neural networks for tracing back to the actual genomic information to try to identify particular markers. I guess COVID-19 and this also gives you a good indication as to what does one want to target, especially for in the development of therapeutics. So those have all been very interesting developments in this area.
Well, thank you so much. Well, we almost covered 1 hour of packed information. We need to process this as we are unfolding, unpacking these information with our audience. Maybe I will request Bridget to kickstart the next phase of Q&A. Bridget.
Yeah. Can I add something to Roderick and Alex's
Sure.
comments, Sushant?
Yeah. Yeah.
Okay,
Karim,
so I'm
please go
finally
ahead.
on the phone. I think hopefully this signal is much easier to hear than my WebEx. So one of the things I was just going to comment on Roderick's point about lung inflammation, that in a sense can also pose challenges in drug development, and how do you actually keep monitoring what's going on with the patients. And typically the way people have done it so far is they've been using CT. And the challenge with CT, of course, is the radiation exposure to the patient is maybe 20 times more than what you would get in chest x-rays. And I'm talking about two-view chest x-rays, right? Like a chest view and a lateral view. So in a sense, trying to use CT to longitudinally monitor patients is in itself posing a challenge, especially if studies have to go on for a long period of time. So that's another place where a combination of AI imaging can help accelerate and potentially refine even some elements of drug development.
Excellent. Thanks, Karim.
Okay, so thank you very much, everyone. And I want to thank the audience as well for joining us, but please stay tuned. The Q&A begins momentarily, and just a reminder to submit a question, click the small Q&A in the bottom right corner of the screen and select all panelists before sending. So a very big thank you to Professor Sushanta Mitra for moderating the discussion today, as well as a big thank you to all of our panelists, Karim, Shirley, Roderick, Ryan, and Alex. Your insights and expertise were extremely valuable. It was a really interesting discussion. I think there's lots more to think about and more we could discuss. Really timely and topical. So thank you very much. For the audience, if you heard anything today that you would like to inquire about, or that intrigued you and you'd like to explore further, I invite you to reach out to me, Bridget Maloney. You can find my email on the Jedi homepage, and or where you registered for this event. And for those of you not familiar with Jedi, it's the gateway for enterprises to discover innovation at the University of Waterloo. So just again, thank you so much to the fantastic panelists and really interesting discussion, and to Sushanta. So I'm just going to show before we move over to the Q&A, I'm just going to share my screen for the upcoming webinar. So I want to show you there that our upcoming webinar, and in fact, our last in the series of 6, will be next week on Thursday, July 25th. The topic is Rebooting with Research, Our Virtual Future. And please go to uwaterloo.ca/Jedi/events for information and registration. And we look forward to seeing you then. So now the fun part, the Q&A, is going to start. And so let me just move over to that. Thanks to the audience. Some questions have come in, and I've been provided with a selection of those that I'll ask the panel. And anyone on the panel, including the moderator, Sushanta, anyone's welcome to contribute to the discussion of the question. And I have the first question. So it comes from the audience via the registration time frame and for this webinar. So Karim, I'd like to direct this first question to you. We're being asked, how can students enter the field of viral detection? Is the first step biology or health sciences degree, and then what? And then maybe you could also comment with from a nanotechnology perspective on that question.
Yeah, absolutely. Thank you for that question. So I think the field is ripe to come into from a range of different angles. Nanotechnology, of course, is a nice field because it blends science and engineering. If you like to have a good handle on the theoretical side and you want to also be connected to implementation, then that, of course, is a great way to go. But absolutely, you could come at it from a health angle, from a health sciences angle, because when you look at a disease, like there's many different facets to it. There's of course the solution like vaccines but there's also things like public policy that is also put into place, right? And if you want to be part of that space, you could come at it from that angle as well, or you could look at it more from an epidemiology perspective, and you could be looking at it there as well. So it really, in a sense, depends on what is really your interest. But in a sense, almost every health profession would have some mechanism of getting in and working on it.
Great, thank you, Shirley.
It's a great question. The first of all, I want to echo what Kareem said. There are researchers from many different fields contributing to COVID-19 testing and vaccine development and etc. It is a very interdisciplinary problem and also solution. Biologists have contributions in one aspect, like study the genomes, developing vaccine, and study the pathologic effects. But I've seen electric engineer, mechanical engineer, chemical engineer, they all contribute. Chemists, of course, in the middle of the diagnostic technologies. Nanotechnology, I had the privilege to teach the students and be involved heavily in the program. I think it's a unique program that students get exposure to a wide range of disciplines. And so that, like Ryan today, they're in a position really to bridge the disciplines and come in to be creative. I think the power of fusing different disciplines is really have a new aspect, a new perspective, to have new ideas and create innovations that may be stuck in one discipline you wouldn't be able to think of even. So I would say many different disciplines will be able to contribute, and you do need to find your own interests and the strengths and have a good heart, and then the rest will be there.
Thanks so much, Shirley. And Roderick, I think you have a comment you'd like to add.
I just wanted to build onto that, to what Shirley and Kareem were saying. I completely agree. I mean, to say antiviral research can mean honestly just about everything or anything. And if you look at just the nature of this panel and the composition of it, and we're all talking about antiviral approaches here. One thing that we found is of course the breadth piece, and for my students, we're not SARS-CoV-2 experts. We all learned it based on whatever our background was, and even on the company front, we had to delve in deep, and just it was a quick and fast approach to learning, being thrown into the deep end. But that breadth piece was the most important. And one thing I always tell even my students is that everything's related to everything. If you don't know how, then you just haven't figured it out yet.
Good point. Very good point. So the questions continue to come in. I'm going to move for question 2. So Shanta, this is a great panel, and you gave everyone a chance to introduce themselves, but didn't have a chance to tell us about the Institute for Nanotechnology and your own work. And so could you do it for us?
Well, when the Waterloo Institute for Nanotechnology is Canada's largest nanotechnology institute, and we have a panel, Kareem, Shirley Tan, Rod Sachet. So these are living examples of how research intense our institute is, and also the undergraduate program, which is run through jointly by the Faculty of Engineering and Science, is also creating the next generation leaders in terms of really helping them to understand the nanoscience and nanotechnology. From my own research perspective, I work particularly in the COVID-19. We are working on the rapid diagnostics, and always give an example. When I was a professor, Ryan was a student, was an undergrad student working on a joint project with Patricia Neva and now he's funding our research. So you have to be always good to your students. That's very critical. And
Comes full circle.
yes,
Very
absolutely.
nice.
And also, we are also looking at from interface science perspective how antiviral coating kind of works in terms of creating surfaces that can destroy, kill, repel these COVID-19 viruses. So these are some of the exciting things happening in my lab as well.
Very nice. Thank you. Thank you for that. So the next question also came in via the registration system. So Ryan, maybe we'll start with you on this one. So any chance we'll ever have in-home testing for this and or other viruses? So it's a multi-part question. And since you're on the commercial side, what are the challenges involved in getting it to the point beyond only the technology required to build an in-home detector? And then sure, you could respond, since it's been the focus of your research too and your company.
Yeah, that's a great question. I definitely think we will see, at home virus testing for COVID and for future viral outbreaks as well. I think the value that level of testing can give you from a public health standpoint and from an economic standpoint, those are some pretty massive forces that are going to be moving a lot of research and a lot of commercialization into this space. It is definitely challenging. Anytime you're trying to take a test out of a lab and put it into a non-lab with people that aren't trained in how to run a proper diagnostic test, things are really challenging. And also to keep it at price points that are accessible. It's all very, very difficult to do, and you have to look at things from a completely different lens than you might normally look at. One example of that is just around supply chain. So, if you look at the scale of testing that you would need to do to contain an outbreak like this really effectively, in the US, for example, you're probably looking at 20 to 30 million tests every day. There is no testing technology available right now that could give you that level of scale without completely depleting supply chains in reagents and materials. So, if you look at pregnancy tests, I think there's 20 to 25 million done in the US per year. So this is a huge scale in terms of the number of tests you'll have to do. So I think supply chain and logistics is ultimately one of the most challenging parts of moving these tests into commercial reality. The technology is definitely one piece of the puzzle that you've got to get right off the start, but you've got to pay attention to these other factors too that will really determine how big of an impact certain types of test technologies can have on the containment of the virus.
I'd like to throw in a comment if possible. What I was going to say is that a vaccine is one tool in our health system's toolkit to address COVID-19, much like how X-ray technology is another tool in the toolkit. We have quite a few tools at our disposal. Some of them are now being used quite extensively. Social distancing, of course, is one. Masks is another. Hand washing, just re-emphasizing the importance of washing your hands is also quite important. It's interesting, the virus has struck across the world, but different health systems and different populations have responded very differently. Some in part because they have better, perhaps they are better positioned to respond well, and others maybe not so. But it's an interesting lesson to learn, right? It's always nice to, it's my opinion, but it's always nice to hope that technology will always solve all our problems. But at the end of the day, there are some behaviors that we also need to keep into mind that probably have maybe even a larger impact. But I did want to throw that in.
Thank you, Kareem. Thanks, Ryan. Shirley, did you want to add anything?
Oh, yes, I think that's a great question. I've been thinking about the at-home testing for the past 5 years, probably. Before the pandemic, it was more general, like how can we just do the testing at home without just going to the lab or without being poked by the needle. Now, when COVID-19 outbreak starts, of course, viral detection becomes the top question for at-home testing. I agree with Ryan that in terms of detection technology, there are many viable solutions in the lab right now, I would say. A lot of activities in research labs, in commercial, the companies are actually ongoing trying to push at-home testing to make them possible. There are, however, hurdles beyond the detection technology. I would name a few. Number one, I would say, is the human factors. How can the user compliance be guaranteed? In other words, when a test is done, how can we trust the test result? Another thing is the cost. Cost is everything. We're looking at probably sending one person to the moon versus sending whoever wants to go to have a tour over there, that type of comparison. And the number three is a much bigger question, is the infrastructure change. We have very centralized healthcare system at this point. To have at-home testing, meaning that the infrastructure needs to be accommodating the distributed model. And in addition to that, regulation changes has to be put in place. But I would be optimistic. I would say some screening tests should be coming our way very, very soon. And advanced technologies are available in the labs. They are being actively pushed across the boundary and reaching the at-home testing probably in the future, not that far away.
Thank you. Thank you. Very, very interesting. Thank you all of you for answering that question. The next question, Alex, this question for you. So the audience member is asking about using AI in assessing social distancing. So how can we strike a balance between the need to monitor using AI and the need to maintain privacy during and after COVID?
Well, that's a great question. So there are many different ways to strike that balance that have been investigated, including myself. One of the key things that we've been heavily using has been more on the computer vision side. So using video streams to assess, social distancing, whether people are, separated, whether they're doing any behavior that might not be appropriate, or the other key thing that has also been of interest has been the detection of whether people have been, wearing PPEs to make sure that at least they're wearing their mask, gloves, so on and so forth, where necessary. So one of the questions here is, how do you actually strike a balance between privacy as well as the importance of doing this? And one of the things that we've heavily investigated is more on the notions of operational artificial intelligence on the edge. And so by having things on the edge, what this allows you to do is allow you to do a lot of the processing directly in a private local manner, directly on an edge device, so that the information that gets sent back and forth is only metadata, so it's de-identified, so that helps with the protection of privacy. The other key thing is also by having it local, what ends up happening is that a lot of video streams can be de-identified, be it through blurring, be it through other ways of mosaicing, so on and so forth, to de-identify the individual. Because what's important from a analytics perspective for social distancing is whether people are separated or not. So it's based on behavior and activity, less in the identification of the individuals themselves. So these are ways that we can do by leveraging more the edge computing, local computing, to do the AI directly on the, let's say, camera devices. This prevents us from having to send a lot of private information and helps protect privacy while still giving us the data necessary to do the proper analytics.
Thank you. Thank you very much. Actually, so the next question is for Kareem, and then the following question we'll direct to Roderick. So Kareem, I'll start with you for this question. While we want to eliminate barriers of bringing research to reality, we also recognize the need to be careful. And for example, we don't want to use our general population as test fields. For example, the Spartan test recall. So what does the panel see as the realistic balance between red tape and acceleration? And within this context, what has our government done particularly well in this pandemic? And are there other bodies that we find are becoming barriers in the deployment of a vaccine?
Absolutely. That is actually a really, really key question, because we know that when a vaccine is deployed, it's going out to a very large population. That's the reason why the FDA has the stage-gated trials, and that's the reason why it takes so long to actually get a vaccine, because you just have to, you have to know that there's going to be side effects. But that also kind of alludes to my earlier point about we have multiple tools in our toolkit, right? We're not just limited. Vaccines would be ideal, but we also know that but maybe it'll happen sooner and maybe it'll happen later. The likelihood seems it's going to happen later because there's still quite a few hurdles to go through. So we have to emphasize the use of what I would term low-tech innovation. And that's what we're seeing now, right? Things like, let's get the masks on, let's get the social distancing, let's get our 2-meter spaces, let's rethink how we do work. Maybe the people who don't need to come into work can work from home. That's the kind of stuff that we have to, as a population, be aware of and be able to execute on. And then just going towards what has our government done well, I think the fact that they moved fast and they got everybody in lockdown mode has helped save a large number of lives. I think the fact that they have a very well thought out plan to get people back to work, which is what we're seeing now across the provinces, is also helping quite a bit. So I think we've definitely done a few things quite well. And are there other bodies creating barriers? It's a feedback system. There's always going to be a push and a pull. And I say it's very valuable to have that, because you need to have checks and balances along the way, because everybody has a valid point, should be heard and then taken into account.
Thank you. Ryan and Roderick, would you like to comment?
Yeah, I just had a couple things to add to that. So I agree with Kareem. I think, obviously we haven't gotten everything right as a country, but I would say that there has been pretty good reception to feedback. I know that Communitech has done a good job sort of surveying, the tech community and CEOs and founders, and understanding like, hey, do these programs help you, or do you not fall into the bucket of eligibility? And if so, how would you recommend, we change some of these programs to make it more helpful for you? So that's more from like, the government wage subsidy programs and things like that. So I think the government has done a fairly good job, modifying those types of programs to better suit the needs of more organizations that maybe they didn't think about in the first place. So that's good. And I also think, we did move fairly quickly with a lot of the public health initiatives, but I would say compared to other countries, it's actually like very slow. I think that's just more of like an experience and like a bit of a cultural thing. I was in Singapore like in the start of February, and they only had like 40 or 50 cases at that time total, but there was like nobody at any restaurants. There was almost nobody out except for like people that didn't live there. There was temperature screening at every workplace, at every single hotel, at a lot of restaurants, hand sanitizer everywhere, masks everywhere. And, they only had 50 cases at the time. So for sure, it took us a lot longer, a lot more cases to get those types of things in place. But I think we also had a lot of cultural barriers to sort of overcome, because, we just haven't really had that type of response needed to public health before.
Right.
Yeah, if I could just build onto that with a quick point, actually, it would be
Yes.
really interesting to look at individualist versus collectivist societies and how they've handled this. We live in a very individualist society where things like privacy are, of course, of critical concern to us. Personalized medicine that we're all used to on our daily basis when times are good. All of this infrastructure is put in place for this type of a culture, which is now flipped completely onto its head. So it's easy to criticize governments for not moving quickly enough, but there are a lot of barriers in place, both cultural as well as infrastructural, to be able to achieve that. I think the most important point, and this is what I'm eager to see and I hope does happen, is that there's a learning component to this so that we can do the learning while the times are good so that we can properly prepare for when things aren't so good, and rather than that short-sightedness where it's over, now let's move on with daily life.
Right. Thank you. Shirley.
Yes, I just want to add to that. On one aspect, we can speed up. We can shorten the development from 12 months to 6 months, from 20 years to 1 year and 2 years in terms of vaccine development. But the other hand, just like Rod mentioned, so we should learn from this crisis. It's not just to address a crisis when the crisis happens. It's rather to have more strategic planning in the system. Solutions and technology development, or even commercialization of a technology, takes a long time. 12 to 18 months is usually how long a pandemic lasts, and that's a very short time frame for anything to happen. Even though we have companies, we have researchers that are working very hard to push out solutions, I would say that most of the solutions will be helpful in the future, but maybe not in time for this particular crisis we're facing right now. So I would say strategic planning and maintain the support to research and the commercialization of medical devices, vaccine development is critical, and that will help us in many years in the future.
Thank you. Great insights on that question. Thank you very much. So Roderick, over to you. Could you comment on the fact that the virus has the potential to evolve quickly? How will it impact efforts to develop a vaccine? And actually, we touched on that, but if you have any additional comments.
Sure. I mean, any virus is going to mutate over time, and this is not. We're already dealing with a virus that isn't the same as originally, was out of China. But in terms of the efforts, it depends on the vaccine is the short answer. So if we're looking to vaccinate, well, then there are different approaches. Some vaccines are very specific toward only antigens or towards parts of that virus that over mutation could be lost. So like flu, for instance, would require subsequent vaccinations over a period of time, which in that case would be a year. What it would be for something like SARS, we don't know yet. On the other hand, multivalent approaches would be ones in which we try to target conserved regions of the virus within a single vaccine. And if that's possible, then that would increase the durability. And what that would mean that hopefully we get some cross reactivity or cross protection from one strain versus another strain over time. In the end, that has to be tested to ensure that that's the case, but there certainly are strategies in place for doing so. And that is certainly something that we've taken into account in the design of our own, is to try to build in that durability.
Thank you. Did someone else have a comment they'd like to add here? Okay, then I'll move on to the next question. I think we probably have time for one more. This one is for Ryan and Shirley. So in general terms, how are you seeing entrepreneurship and commercialization help bring lab-based research to the market? So maybe could you talk about how it happened for you?
Yeah, definitely. So, yeah, I think that it's really important that there are good programs in place to support entrepreneurs. And I mentioned this earlier with like the Velocity program and some of the other ones in the KW area. But if you're going to start a company and try and commercialize a technology coming out of a research lab, there's no question that like you're going to be a total expert in that field, right? Like you're going to know the technology inside and out, and that's really great. But you also have to understand all the other parts of building a company, and that's really hard. And it can take a really long time and lead to a lot of costly mistakes if you don't have people helping you out at the beginning. And so as much as I think, as much as scientists and engineers often think that the technology is the absolute most important part of the company, there are so many other factors that come into play that affect whether or not a company will be successful, and therefore, a commercialization effort of a technology from a lab will be successful. So it's really important to not get just fixated on only one dimension of the business, which is where we tend to go because it's where we're comfortable with, right? We understand the technology, we feel confident talking about it. Maybe you don't know anything about finance and you are really intimidated by balance sheets and P&Ls. So but it's still important to be like, okay, and accept that you don't know that stuff and just like find the support and help you need to do it, because all these things are important to being, successful in bringing a technology out of a lab. And there are many avenues to be successful with doing that. The one I mentioned is really just one, which is, starting your own company, but there's other ways of doing it, whether it might be like licensing it to, another company to commercialize and like working with them on it. I think there's lots of good opportunities to do that too. And in many cases that does make a lot of sense, because you can leverage something that a company is already really, really good at. But in practice, those are not always the easiest things to do, because you really need to have good relationships with typically large companies that are challenging to get momentum within. And at the end of the day, the person that has the most at stake is usually the one that had invented the technology and came up with the technology. They're going to be the most passionate about it and the most likely to weather the various storms and challenges that they'll face when they try and commercialize it, which is why I think that is the most likely route to being successful with these types of things.
Thanks. That really good points in there. Shirley.
Well, I would say that entrepreneurship and starting a company is definitely a lot of work. There's certainly a death valley between you start a company and the success of the company. That every one who's thinking about entrepreneurship should be prepared that death valley is on the way, that you have to somehow manage to get over with it. To me, I speak as a researcher and a scientist. I see entrepreneurship as a need that come up over the time. Because when I initially as a scientist, we would say, okay, the fundamental science is in place, the technology is ready, I'm done. But then when I talked to doctors and the real world application or the users, there's a huge gap between the technology and the real world product. And that's when I actually jumped into this entrepreneurship and tried to actually help with commercialization. And then I find out that really the boundary has to be pushed. From the science, I have to go into a system solution, which I have to deal with a lot of engineering aspects. And then the user factors come in, the communication, the commercial aspects, like how you market it, how you bring to the user, and who will be the user, what is the specific questions that your product will try to address, and also how do you produce reliably a product? All those questions come up. So it's exciting, and many facets involved.
Exactly. Thank you so much, everyone. I'm so sorry we're out of time. We did have a couple of more questions, but we are actually a little bit over time. So I want to thank the audience very much for your participation, but a huge thank you to Sushanta, Shirley, Roderick, Alex, for answering all the questions and you know, it's been such a topical and timely discussion, and it was really very informative and enlightening. So thank you very, very much. Again, if anyone in the audience has heard something and they'd like to explore further with any of the panel or anything else at the University of Waterloo, please reach out to me, Bridget Maloney. And just a reminder, our next and very last panel of our 6-panel series is next Thursday, June 25th. And the topic is Rebooting with Research, Our Virtual Future. And go to uwaterloo.ca/Jedi/events for information and registration. So thank you very much, everyone. We look forward to seeing you next week to the audience. And this closes our session for today. So have a great day, and thanks for joining us, and take care.
Thanks very much, Bridget. Thanks.
Thank
Thanks,
you.
everyone.
Bye.